Oh, it's time to start, isn't it? It's already 4 o'clock. Well, welcome to another episode of Seminar in the Time of the Coronavirus. We've got a great lineup this semester. We've got a few less students than we expected, I guess. And we do still have a few slots. So if any of the faculty out there are interested in presenting a seminar, just send me a note and we'll put you on the roster. I won't waste any more time. We've got a great speaker today. I'm going to turn it over to Dr. Gentry to introduce our first speaker of the series. Go ahead.
Thanks, Scott. It's a pleasure to introduce today's speaker, Dr. Katie Lewis. Katie was raised on a farm in Taft, Texas. It's down in the coastal Bend area, barely above sea level, so very different than her current position in Lubbock. She got a bachelor's in chemistry from Sam Houston State University and then came to Texas A&M to work on a master's with Frank Hans and me on the effects of oilseed meals on the germination, growth, and survival of crop and weed species. And then she did her Ph.D. in soil science at A&M with Jamie Foster, working on soil and plant responses to lipid-extracted algae soil amendment. And then she started her current position as an assistant professor, soon to be associate professor of soil chemistry and fertility at Texas A&M AgriLife Research in Lubbock, where she also has a partial appointment at Texas Tech teaching soil fertility. And Dr. Lewis has built a very strong research program that you'll hear more about shortly. And notably, she received the J. Tom Cothran Outstanding Young Cotton Soil Scientist Award from the Beltwide Cotton Conference in 2019. So being recognized for some of the outstanding work her program is doing. And with that, it's my privilege to turn over to Dr. Lewis to hear more about her research.
Thank you, Dr. Gentry, for that wonderful introduction. It is kind of crazy to think that the last time I presented at seminar was as a PhD student about 7 years ago. So time has definitely flown by. And thank you, Dr. Finlayson, for the invitation to present here today and to give you, my goal is to give you a good idea of what we do within my research program. And the way that I plan to do that is to give you a snapshot of my graduate students' research projects. So I think it's important to start out by talking, well, if I can get the slides to move. There we go. All right, so important to talk about who is involved with this research. My program entails research associates, technicians. I have multiple graduate students, some of whom you know who are on Texas A&M's campus, but as Dr. Gentry mentioned, I also have an appointment at Texas Tech University where I have several graduate students as well. And it also includes undergraduate and high school student assistants. This doesn't list here all the many collaborations that I have across the state, from Corpus to Vernon and on campus as well. And I'll mention those throughout the presentation today. So what we do, of course, I'm going to go into a lot more detail, but to give you just a brief little overview, our focuses really can be divided into three main areas: nutrient management, cropping systems, and soil health. And the majority of this research is being conducted across the southern high plains of Texas. Like I said, I have many collaborations across the state of Texas and even throughout the Cotton Belt. But from our standpoint, most of our research is being conducted on the southern high plains that you can see here. And we do have some other projects that go more into the Amarillo area, even north of that, into more of the high plains. So a little background on the southern high plains. And some of you may have been wondering what the heck I was talking about when I said soil and nutrient management at 3,200 feet. Well, Lubbock sits at 3,200 feet, nearly 1 kilometer above sea level. And like Terry mentioned, much different than where I grew up in southern Texas that was at sea level. This area of Texas is encompassed by the MLRA 77C, which you can see here, the darker orange color, and it encompasses about 54,000 square kilometers. And it is part of the US Southern High Plains, and we are in that southern portion. Across the southern high plains of Texas specifically, we have pretty large diversity as far as soil textures. We range from clays and clay loams in the northern part of the high plains. and we go down to the southern part where we have much more sandy textures. And so this gives us an opportunity, especially in the Lubbock region, to really get a good variety of soil textures. We can place projects across this region and get different responses from that textural component. The predominant soil orders that we have are alpha soils, which include the Amarillo fine sandy loam, which the majority of the research that I'll be discussing today is conducted on. We also have mollisols and septosols and some iridosols as well. As far as the climate goes, again, very different than what I grew up and even there in College Station, where we have an extremely arid climate, with our temperatures ranging from the northernmost part down to the southern part of the southern high plains from 13 to 17 degrees C on average. This is our annual average. Our rainfall ranges from 405 to 560 millimeters. Our wind speeds, which can cause some major issues on the southern high plains, on average are about 20 kilometers per hour. And as far as our frost-free days, that can range anywhere from 195 to 255 days per year. Let's move down to the figure where I have temperature, precipitation, and wind speed, because this is really important when we consider our growing environment that we're dealing with. We are planting cotton, which is our primary cash crop in this region, in early May into early June. And if you notice, that is our area when we have the lowest precipitation and the greatest wind speeds, which makes it extremely difficult to get our cash crop, our cotton crop, established. Another challenge that we face on the southern high plains is our evapotranspiration, or potential evapotranspiration, where our average annual PT exceeds the precipitation by 2 to 3 times. So this would be the reason that over 50% of our acres, cotton acres, are irrigated, because we have to depend on that irrigation. Dry land production ranges from 40 to 60% of our total acres in a given year. And it's definitely a challenging environment to make dry land production work. So with those sandy soils, with the dry climate, with high wind speeds, we have high potential for wind erosion. And this dates back to the Dust Bowl, 1935. This is in Hansford County, quite a bit further north in the Lubbock region, but it's definitely not something that's new to the area. I like to joke, the picture on the right was a picture that was sent to me by my mother-in-law prior to Clay and I moving up to the Lubbock area. And I like to joke that I didn't think she really wanted us to move up to this area, because otherwise, why in the world would she send me a picture of the dust blowing like this? You know, we have done a lot since the Dust Bowl. We've reduced our wind erosion by about 40%. However, it still happens. We still see this wind erosion, haboobs happening each year. And so that's why it's really important for us to continue to try to optimize our cropping systems, implementing cover crops, different irrigation strategies in order to try to reduce the amount of wind erosion that we have in a given year. So before we get into the research, I think it's also important for us to understand the timeline of crop production, specifically cotton, on the southern high plains. Because of course, just like anywhere, if we move across the cotton belt, our planting dates are going to be adjusted, harvest dates, and then how that influences our cover crop season. So in a given year, we're going to plant cotton sometime between May and June. Early June is usually our deadlines for planting. We're going to harvest starting early November into that December timeline. This is one of the challenges of making a cover crop work in this region, is because we have a limited amount of time to get the cover crop established before then having to terminate it in mid to late March, maybe even into early April. We just don't have that length, those warm days, to really get a good amount of biomass produced from a cover crop. Moving on to the research. What I'm going to discuss today can be divided up into really 5 different studies. So the first one is going to focus on cotton nutrient partitioning. I'm really excited about this research. We recently have it published, and I think you'll find it interesting as well. Moving on to cover crop management. including seeding rates and termination times. Also looking at cover crops, cotton, and how that all relates to soil health. Another project is going to be involving cover crops as well, but adding a nitrogen component into that and how that all influences greenhouse gas emissions and soil biology. And then finally, the newest project that we've started is an organic cropping system, a rotation of cotton and peanuts, and how both organic and conventional systems influence soil health. And so with that, I'll jump right into that research. As I mentioned, the first project that I'm going to discuss is focused on cotton nutrient partitioning. So from the time that I started here in Lubbock, which would have been almost, I guess, September 2nd will be 6 years. I just had, reading publications, looking at other research that's been conducted, not just here in Lubbock, but across the Cotton Belt as well, started realizing that we've had so many changes in cotton genetics over the past 30 years. But we hadn't really done much from a nutrient standpoint as far as looking at how those nutrients are being partitioned within the plant. And so that was one of the things I tried for several years to get some funding. Finally, Cotton Incorporated funded the project, and like I said, we recently had a publication from this research. And I'll go through some of the major findings from it. First one, and I think it kind of leads into the other findings, was the changes in resource pools and how carbon is being allocated throughout the plant throughout the growing season. And we're comparing all of our research back to research that was conducted in the early 1990s. Mullins and Burmeister is the report or the publication that we're comparing back to. And so comparing back to that, one of the first findings is that newer varieties or more modern cotton cultivars have become more efficient at partitioning their dry matter into the reproductive structures or parts of the plant. And so what I mean by that is if we look at the percentage of total dry biomass that is being produced, there's a larger percentage of that in our newer cultivars that's being partitioned into the bolls. So the seed, the lint, the reproductive structures of the plant. And with that, that's going to equate to greater lint yield with less vegetative biomass being produced. And here at the bottom, the table, we have our 3 different varieties that we were looking at. And to introduce those, Paymaster HS26 was a variety that was extremely popular in the 1990s. Fibermax 958 was early 2000s, and Delta Pine 1646 has been the variety for the last several years that's been planted on the greatest number of acres across the Cotton Belt. And so we really have a good comparison of varieties from the 90s to current. And what we've seen is with that increase or more efficient partitioning of dry matter to the reproductive structures, we've also seen an increase in lint yield in the 2 years that we've conducted this study. And with that shift in dry matter production, we also see a shift in nutrient partitioning as well. And I know there's a lot going on this slide, but the main takeaway from this is that our more modern cotton cultivars, so Delta Pine 1646 and Fibermax 958, have become more nutrient dense in those reproductive structures. So they're moving more of their nutrients into the seeds and the burrs of the plant. And if we look specifically at nitrogen and phosphorus, when we talk about the amount of those 2 nutrients that are being partitioned into the seed and comparing that back to Mullins and Burmeister, we ranged anywhere from 53% to 63% of the total nitrogen being partitioned into the seed compared to Mullins and Burmeister, who was at about 42% is what they reported. So large increases in the amount of nutrient that's being partitioned into the fruiting structures. And that can also be seen with the total amount of nutrient that's been taken up, and then also looking at the nutrient index, which is the amount of lint yield produced per unit of nutrient taken up. And this is comparing back to the Mullins and Burmeister report. So we have a percent increase here on the table. With nitrogen, for example, total uptake increased by 36%, and the lint yield produced per unit of nutrient uptake increased by 66%. Phosphorus also increased quite dramatically as far as the nutrient index is concerned. And so with this data, we may be able to make some adjustments as far as how we are managing these nutrients in a cotton crop. So moving on from that, the next study that I'm going to discuss is looking at cover crop management. Oh, sorry. Cover crop management. And this research is conducted by Ray White, who is a PhD candidate at Texas Tech University. He is co-chaired by myself and Dr. Wayne Keeling. And his project has two components. The first one is the one that I'm involved with, and that's looking at cover crop seeding rates and termination timing and how that affects biomass production of the cover crop, as well as how it influences cotton yield. His second component is more weed management, which that's Dr. Keeling's area of expertise. What he was looking at was, again, comparing seeding rates and termination timings, because that's a question that we get quite often from farmers in the area is, should I be planting wheat or rye as my cover? When should I be terminating it? And how much seed should I be planting? And there are recommendations from NRCS, but we thought that it'd be a good idea to evaluate that ourselves and look to see if we couldn't save a little money from the standpoint of seeding rates, but also to see when the best time to terminate that cover crop is. And so this data is from 2017 to 2019, very similar trends between each of the individual years. If we look here on the left at 2017, we have pounds per acre on the y-axis, and then our different cover crop treatments along the x-axis. We'll focus first on rye, and the trends between rye and wheat are very similar, but with rye, there was no difference between seeding rates between the 30 and 60 pounds. Where we did see the difference or the separation in the amount of cover crop biomass produced was between the optimum and late termination time. And the difference between the optimum and late is just a 2-week time period. So optimum would be based on NRCS recommendations, and then the late is 2 weeks after that. And it's just amazing to me to see that large increase in biomass production from the optimum, waiting 2 weeks and then terminating. And that same case is with the 60 pounds, whether or not it's rye or wheat that we're using as our cover crop. Very similar trends in 2019. However, in 2019, we were extremely dry and did not get near the amount of biomass produced in 2019 that we saw in 2017 and 2018. And what's most interesting is to see how this relates back to cotton lint yield. Because one of the things that you hear quite often with cover crops is that you need to produce a greater amount of biomass to help control weeds, to see the greatest benefit from a soil health standpoint. Well, when it comes to lint yield, what we have seen, and we have lint yield, cotton lint yield on the y-axis, cover crop biomass on the x-axis, and then each of our 3 individual years that are represented. 2017 and 2018, what we see is with an increase in biomass production, we see a reduction in lint yield. 2019 was that strange year where we really didn't see much variability in the amount of cover crop biomass produced or in our lint yield. So we didn't see that same relationship that we see in 2017 and 2018. And so the takeaway from this, and this is something that we present quite often at grower meetings, is that number one, you can save money by planting half the seed. So plant 30 pounds rather than your 60-pound seeding rate. And then also make sure that you are terminating that cover crop in a timely fashion so that you don't get that additional biomass produced and see that same reduction in lint yield. And hopefully with the next set of data that I present to you, we can get a better idea of possibly what's going on. Why are we seeing that reduction in yield with the increase in cover crop biomass? So the next project is going to focus on cover crops and how that relates to soil health. And this project is led by Joseph Burke, who I'm sure many of you know. He's a PhD student at Texas A&M in the Soil and Crop Sciences Department. And his research has many objectives. The ones that I'm going to focus on today are first to assess soil health metrics in semi-arid cotton production and determine the relationship between cotton yield and soil health. And then also to quantify water availability following cover crops in a cotton production system. So again, the area that we're working in is in the Southern High Plains in Dawson County, 2 counties south of Lubbock. It is at the Ag Cares Farm, or the Agricultural Complex for Advanced Research and Extension Systems. It's a cooperation between Dawson County cotton growers and Texas A&M AgriLife Research and Extension. And in 2018, working with Dr. Paul DeLon and Vernon, we received a contract with NRCS to evaluate the effect of long-term systems on soil health parameters and compare that back to a native system. And so we searched far and wide to identify a location, a native site in the same area with a similar soil texture. And working with NRCS, we were able to identify one of these locations in Wellman, Texas, which is about 60 miles as the crow flies from that Lamesa long-term site where we have been conducting research for the last 22 years. And so the systems that were being evaluated, we have a continuous cotton system that was established in 1998 with 2 treatments at that point in time. There was conventional tillage, winter fallow, and then a no tillage with a rye cover crop. And remember, these are continuous cotton systems. So cotton is planted every year in that May timeframe and harvested in November. The conventional tillage winter fallow does not have a cover crop versus the no tillage with a rye cover. We'll plant the rye cover shortly after harvesting cotton and then terminate it in that March timeframe. In 2014, right when I first started, thanks to Dr. DeLon, he received an NRCS grant, and we were able to continue these plots and add in an additional treatment. There was a mixed species cover that includes hairy vetch, winter pea, rye, and radish. And so we have these 3 systems at our Lamesa location. And then for the research I'm going to be discussing, we also have this native site that, according to the NRCS, their historical records had not been plowed for at least 80 years. So first things first is to look at cotton lint yield and those 3 systems that I mentioned. So we have conventional till in the gray bar, rye no-till is the green bar, and mixed species no-till is the blue bar. And we're looking at cotton lint yield from 2015 to 2019. And of those 5 years, we've seen a significant reduction in lint yield where we have a cover crop in place compared to the conventional till system, and specifically where we have a rye cover crop. The mixed species cover crop in those 2 years, we have not seen a significant reduction in lint yield. In 2019, we saw similar trends to what we saw in 2016 and 2017. So the question is, what is causing this yield drag that we see in some years? And going back historically, prior to me starting here, Wayne Keeling has those records, and generally we do see a reduction in lint yield with that rye cover crop compared to the conventional till system. And so there's several different possibilities. The first one that you hear from most people is that it's a water usage issue. So we have our cover crop in place. That cover crop is utilizing water just like the cotton crop will. And so we have a deficit starting out early in the season. And then the second possibility that Joseph is investigating is looking at nutrient immobilization, specifically looking at nitrogen. With a large amount of cover crop biomass that we're leaving on the soil surface that's decomposing throughout that growing season, how much of the fertilizer nitrogen that we're applying is being immobilized by the microbes as they're decomposing that cover crop material. So first we'll start with soil moisture. And once again, another busy slide, but I think it tells a really nice story. So what we're looking at is soil moisture in millimeters in the top meter of the soil profile. And then along the x-axis, we have days after planting, and that's planting specifically the cotton crop. And so where we have these negative values, -53, -22, that would be 22 days prior to planting our cotton crop. Same color scheme where conventional till is in the gray lines, rye no-till is green, and mixed species no-till is blue. And what we generally see in each of the years is we see a reduction in soil moisture prior to planting our cotton crop. So from the time that we plant the cover crop to when we terminate it, that cover crop is utilizing soil moisture and creating this deficit where we have the cover crop in place. And we see that in each individual year, 2015, 2016, and in 2020, it was very dramatic where we have a large reduction in soil moisture where we have the cover crop in place. But one thing you'll notice is as soon as we have either a rainfall event or we're able to irrigate, that moisture deficit is made up. And there's really very little difference in soil moisture throughout the growing season between those three different cropping systems. And even in some cases, we do have greater soil moisture where we have the cover crop in place compared to the conventional till system. There's lots of advantages to the cover crop. It's reducing evaporation losses. It increases water capture and water storage. And so we're seeing that throughout the growing season. And based on this data, 6 years now, we can confidently say that it's not moisture that is limiting our cotton yields. And so there's got to be something else going on. Like I mentioned, Joseph is also looking at nitrogen dynamics, nitrogen immobilization, what's available to the cotton crop throughout that growing season. And hopefully that's something that he'll be able to discuss with you in next semester seminar. Moving on to the soil health parameters that we've evaluated in the system, looking at soil carbon, both as organic carbon and potassium permanganate oxidizable carbon. The figure on the left is soil organic carbon. And along the x-axis, we have two depth increments, 0 to 5 centimeters and 5 to 10 centimeters where those samples were collected. And with LSD letters, we're looking at differences within that depth across the different management systems. I've also included the native site here as a comparison, because a lot of the times when we're talking soil health, one of the things that people are striving for is to get back to those native systems, to move away from managed systems, to have more of a natural system, because they believe that that should improve the overall health of the soil. If we look first at soil organic carbon and compare these different systems, there's no difference between our managed cover crop systems and our native site, but all three of those systems are significantly greater than organic carbon in the conventional till system. And the same holds true, at least for the two cover crop systems compared to the conventional till system in the 5 to 10 centimeter depth. The difference at this depth, though, is that we're seeing greater organic carbon where we have that managed cover crop compared to the native site. And the idea behind that is in that managed system, we are adding large amounts of organic material to the system. Our inputs of organic material from the cover crop are much greater, both above and below ground, compared to that native site in a given year. Trends are very similar when we look at potassium permanganate oxidizable carbon, which this is an estimate of the active carbon in our soil, where we have greater active carbon with both the cover crop systems compared to the native and the conventional till at both the 0 to 5 and 5 to 10 centimeter depths. Another measure that is an indicator of soil health is enzyme activity. We looked at both beta-glucosidase and beta-glucosaminidase, which beta-glucosidase gives you an idea of carbon mineralization. The beta-glucosaminidase is both carbon and nitrogen mineralization. Very similar trends to what we looked at with the organic carbon and active carbon, where our managed cover crop systems result in greater enzyme activity compared to the conventional till at both depths and both enzymes. The one difference that you see between the beta-glucosidase and beta-glucosaminidase is the difference between the two cover crop systems. So in the case with beta-glucosidase, we see generally greater enzyme activity with the rye cover crop, whereas beta-glucosaminidase activity is greater where we have the mixed species cover crop. And if I was teaching my class right now, of course, I would ask all of you why that would be. And it goes back to the idea that the mixed species cover has a legume in it. So we're adding that additional nitrogen to the system and most likely enhancing that activity from the beta-glucosaminidase. Again, big question. How does all of this biology related data, how do, at the end of the day, when we're talking to farmers, yield is going to be one of the most important factors because they have to continue farming from one year to the next. And we're improving all of these biological parameters in the soil, but how does that relate back to yield? And if we look at a principal component analysis, there was not a significant relationship between yield and any of the measured parameters, the biological parameters that we're measuring. And there's a much larger list of these, and I'm sure, again, once Joseph presents this at seminar to the department, he'll include those in his findings. But I think it's very interesting that we can see such great improvements, large improvements in our biological parameters of the soil, but yet it's still not being reflected in yield. And that's something we're working on very diligently, is to try to determine exactly what it is that's going on with the cotton crop. Is it, like I said, a nitrogen limitation, or is it something related to disease or nematode? And that's something we're working on with our plant pathologist here in Lubbock. The next study is going to be one focusing again on cover crops, but also involving a nitrogen component to that. And this project has been led by Mark McDonald, also a PhD student at Texas A&M in the Soil and Crop Sciences Department. And he is co-chaired by Terry Gentry and I. And sorry, I forgot to mention, so Joseph is a graduate student of both mine and Julie Howe's. So with Mark, his research is definitely more biology focused, and I know he's presenting at seminar this semester, so the results that I share with you from his research today are going to be more limited than what I shared of Joseph's. Mark's project, he came to me as a master's student, did his master's project research through Texas Tech University, and so his PhD work is a continuation of that. He had really good questions that were generated from his master's research that he wanted to continue forward through his PhD research. And the two objectives from his dissertation that I'm focusing on today are focused more on greenhouse gas emissions and how cover crops can influence or help mitigate the emissions from those systems. And then also looking at how the above ground management practices can influence cotton yield. So his study is conducted in Lubbock at the Research Center. Different soil series here at the Lamesa location that I was just discussing. That's in Amarillo Fine Sandy Loam. Here in Lubbock, this the soil here at the center is a ACUF or Olton clay loam. And Mark has two locations, not just here in Lubbock, but also in Vernon. Paul DeLon is also involved in Mark's research. These projects were initiated in 2015, and it's a split plot design where we're looking at no-till with a wheat cover, no-till winter fallow, and then a conventional tillage winter fallow. And then the split plot in this design is nitrogen fertilizer timing. Of course, we have a control with no added nitrogen, but then we have different timings of that application. 100% pre-plant applied, 100% in-season side dressed applied, and then we have a split application that's 40% pre-plant, 60% side dressed, and then a 100% pre-plant with a stabilizer, nitrogen stabilizer product added in. First thing, lint yield. Very different results than what we've seen at our more sandier locations. And that's a trend that we've really started noticing in this cover crop research, is the soil texture effect that you're going to see from south to north as texture changes. But here in Lubbock, Mark's study started in 2015. So we have lint yield from 2016, 2017, and 2018. And we have no-till wheat, that's the black bars, no-till winter fallow in the gray, and conventional tillage is the white bar. In the first year of the study, so we had implemented the cover crop in 2015, planted cotton in early 2016. So this is 1 year after a cover crop was added into the system. And we see similar trends to what we see at the Lamesa location, where a conventional till yields more than the conservation systems, no-till fallow and no-till with a cover crop. However, in 2017 and 2018, we see a change in that trend with the cover crop, the no-till wheat system, yielding more than the conventional till system. And this holds true for both 2017 and 2018. And the idea behind this is that with that cover crop, it offers protection to cotton seedlings that are not offered in the conventional till system, where you can have wind erosion and sand damage to a young cotton crop. Looking at the CO2 emissions, both CO2 and N2O are measured using a GasMet FTIR. So measurements are measured in, or gases are measured in real time out in the field using the GasMet instrument. The data I'm presenting today is from 2016 here on the top and 2017 along the bottom. And then the difference as we move from left to right is an average of those fluxes from the spring, summer, fall, and winter timeline. And then we have the same color scheme for the no-till wheat, no-till, and conventional till. And what we see with CO2, we're gonna have greater fluxes, or at least differences in those fluxes in that spring, early summer timeline, with the no-till wheat system having a greater CO2 flux rate compared to those other two systems, which makes sense. We're getting greater soil respiration because we have an actively growing crop in the ground during the spring time frame. And in the fall winter, we're getting our cover crop established. And again, we see a greater CO2 flux rate with the no-till wheat system. However, even though we're seeing greater CO2 emissions, because of the carbon that's being assimilated by that wheat cover crop, we have less net losses of CO2 in the no-till wheat system, which should over time result in greater carbon storage from those systems because of the carbon that's being assimilated by the wheat cover crop. And with N2O emissions, we don't see a difference in cropping system. Where we see the differences is going to be related to nitrogen management. And again, we have our flux rate on the y-axis with the different seasons along the x-axis. And our greater N2O production is going to be in this summer season, summer time frame, where we have warmer soil temperatures and more microbial activity are going to increase the amount of N2O that's being lost from the system. And I don't know about you, but the first thing that I really noticed when I look at this is these negative values here. And that's something that Mark started noticing during his master's research. And I actually went back and looked at some of my PhD work, and we also had significant negative fluxes. And what you'll notice, it's going to be predominantly in nitrogen limited environments. So here in this summer season, in both 2016 and 2017, where we have a negative flux, is going to be where we are adding, where we're not adding any inorganic nitrogen fertilizer. And then by the time fall, winter comes around, all these systems should be more limited in nitrogen fertilizer just because of the acquisition of that nitrogen by the cotton crop. And so again, we have negative fluxes. So this is one of the big questions that got asked from Mark's master's research, is what's going on? This is something that is cited throughout literature, the observation of negative flux of N2O from the soil surface, or consumption of N2O within the soil, but no one has really been able to determine exactly what it is that's causing this consumption of N2O. So Mark's research is focused more on the biology for his PhD work and looking at trying to identify what microorganisms are responsible for the consumption of N2O from our agricultural systems. And the final project is actually one of the newest projects that we started last year, and it's in collaboration with Paul Dewan and Terry Gentry. And the graduate student that's leading this project is Leah Elman-Storch. She's a master's student at Texas A&M in Soil and Crop Sciences Department. And the objective of this project is to investigate how cover crop choice, whether or not it's a single species or a mixed species, impacts soil health through both microbial activity of agricultural plots that have recently been transitioned to organic management systems. And so we're comparing these organic systems to a conventional production system. The locations again include both Lubbock and Vernon, and it's a split plot design. Planted to cotton peanut rotation. The cotton was planted in 2019 and will be planted again in 2021. And we have peanut in rotation with the cotton that was planted earlier this year in 2020. We have several different cover crop treatments, but there are 4 that are similar between both the organic and the conventional production systems. Here we're looking at carbon mineralization, which is one of the more common soil health parameters that's being measured. On the left, we're looking at the 0 to 10 centimeter depth, and on the right is the 10 to 20 centimeter depth. And on the x-axis, we have our different cover crop treatments. So we have rye at 30 pounds per acre, radish at 10 pounds per acre, a rye vetch mixture that's a 30-pound seeding rate, 25 of that being rye, 5 pounds of that being vetch. And then we have a 3-way mix that's rye, vetch, and radish. And again, a total of 30 pounds of that being seeded with 25 rye, 3 vetch, and 2 radish. And the major differences, if we compare the organic system, which is the blue bars, to the conventional system, which is the green bars, where we see the biggest differences is in the rye at 30 pounds, with the conventional system resulting in greater carbon mineralization than the organic system. And then with the rye vetch mix, again, the same trend with the conventional till having greater CO2 losses or carbon mineralization compared to that organic system. And the next aspect of this project is looking at farmer fields. So we have 4 different farmers that we are working with to look at the effects of organic production systems and how that compares back to a conventional system. And we're planning to measure a whole slew of different soil health parameters, but one of the ones that I found to be most interesting was looking at phospholipid fatty acids. This gives us an estimate of microbial biomass in the soil and comparing the organic systems in the blue bars and the conventional system in the green. And a lot of the times, especially when you talk to some of the people within the organic production systems, is that they believe that organic production is going to be better for the environment. It's better for the overall health of the soil because all the pesticides that we're adding in a conventional system is going to be detrimental to the microbial biomass and what's going on below ground. And so these results really were exciting to me. The brown system that's here on the left has been in place for at least 10 years, and the conventional till system has greater PLFAs compared to the organic system. The Warren farm is much newer. It's less than 5 years that the organic system has been implemented. And here it is significantly less with the organic system compared to the conventional system. And the big difference between these two, organic production relies heavily on mechanical weed control. So lots of plowing, lots of tillage events are occurring in an organic system, whereas in a conventional system, they are relying more heavily on pesticides, but they also are more successfully able to implement a cover crop into the system. And as we've seen with previous data, where you have additional organic material inputs into the system, that's going to influence the microbial activity in the soil. And so I believe that's exactly what's going on here, is we have a cover crop in the conventional system compared to winter fallow in the organic system, where we're doing more plowing, disturbing the soil, and we have less microbial biomass in those organic systems compared to the conventional systems. So that wraps up the individual research projects. Just to give you a little overview of the other research that we're doing, I was recently funded to start doing some fertigation work, looking at how we can manage our nutrients via fertigation in a subsurface drip system and in cotton. We're also looking at that nitrogen aspect and how it relates in a cover crop system, how it's influencing nitrogen availability and plant uptake. We also do some on-farm evaluations with cover crops and crop rotations in corn, and that's more in the northern panhandle, working with Jordan Bell and Dana Porter on that research. And then recently, Paul and I have started working on calcium management in peanuts. That's an issue that quite a few of our peanut growers have in this part of Texas, is they believe that they're having some calcium limitations, even though our soils are loaded in calcium. And then finally, evaluation of different soil amendments and fertilizers is something that we also work on. And with that, I had to end with a picture of my greatest achievement of 2020. Cadence was born in March earlier this year, and she has just been absolute joy to both Clay and I. So I had to share some of that joy with all of you. Thank you for all of your attention, and I would be happy to answer any questions or comments, address any comments that you have.
Good job, Katie. Great job.
Thank
Fire away. Hammer her with questions.
Okay, I do have a question. Earlier in the introduction, it was said that the modern cultivars, they have like better efficiency on the allocation of the resources to the reproductive system, so than the old cultivars. So can this be attributed to breeding of better, better yield, or is it more to the manipulation of the crop physiology and the cultural practices?
So I'm not sure I'm the best person to address that question, but I believe that it would be more related to the breeding aspects that have occurred, the changes that have occurred over the past 30 years. I don't know if any of our breeders are online or not, but and that was always the idea or the hypothesis, is that we've had changes in our genetics over the past 30 years that have influenced yield potential and water use efficiency of the crop, and so the nutrient dynamics had to have also been influenced or changed through those changes in genetics.
All right, thank you.
Thank you.
So Katie,
Yes, sir.
I'm aware that there are frustrations occasionally with being a new assistant professor and going through the process.
You're cutting out. I didn't hear you, Dr. Baldsberg.
So what's been your biggest frustration with your job and your role?
Oh, wow. Put me on the spot. My biggest frustration.
Besides me, of course.
Yeah.
And frustration, how you've dealt with it to go on and be successful.
Yeah. So the first thing that comes to mind is so for example, the nutrient partitioning work that we've done. That was something that I recognized as a gap in the literature over the last 30 years, but it was really difficult to get the funding to be able to conduct that research. It was something that I had to work pretty hard at and do some convincing within Cotton Incorporated. But being a soil scientist working specifically in cotton, there's funding limitations associated with that. So I feel like I've had to go somewhat out of my area of expertise and really be more flexible as to what research we're conducting. I'm sure I could come up with several other things too if you gave me the opportunity. But funding challenges has definitely been a big one. But I think it was Dr. Cawthron that told me way back when that sometimes, and I'm not going to use the exact words that Dr. Cawthron said, but sometimes you just got to do the things that you don't necessarily want to do to be able to do the things that you're really interested in.
Well
Katie,
said.
sorry.
Yes, sir.
Katie, may I ask you one of knowledge question? You mentioned several things. Something I'm wondering what kind of roles minerals play in there. For example, you mentioned calcium issue, and earlier you also mentioned some nutrients. Sometimes the cotton responds to fertilizer, sometimes they don't. I'm wondering, what's your opinion about how those minerals affect those kind of efficiency or calcium deficiency?
So one of the projects that I didn't discuss was some work that I did early on looking at potassium dynamics in cotton. And we see a response, and this is something that's noted across the cotton belt too, where we see a response to added potassium fertilizer, even though our soil test values are 2 times greater than what the critical level is. And I believe that a lot of that goes back to the mineralogy of the soil and the potassium fixation potential. If Dr. If Tony's still on, he's going to argue with me that it's more related to roots, which I do agree to an extent with that. But I think the mineralogy does play a major role in what's going on with potassium and most likely calcium as well. I'm not sure that necessarily all the calcium that we believe and we have in our soils is truly plant available.
Okay. So for the fertility test, normally people take one sample and then get the value. I'm wondering if the sampling time will be important here or not, such as in the winter or in a dry season or your rainy season, will that affect that numbers or not?
Oh, most definitely. And we haven't looked at that specifically from a nutrient standpoint, but we have, part of Joseph's master's work was looking at temporal changes in carbon and nitrogen throughout the growing season. And with those two specifically, in nitrogen, we know, of course, is going to be extremely dynamic. But with carbon, we were seeing some major differences, and it really followed the pattern of what we would suspect photosynthetic rates would be of cotton, where we were seeing greater organic carbon in the soil in that July-August timeframe. And so I think that would also influence your nutrient dynamics, especially those that are going to be influenced by microbial processes below ground. But also, like you said, dry, wetting, that's going to influence it too.
Great, thank you.
Thank you.
I have a very simple question for you, Katie. I was curious because I'm not a soil scientist and I've never worked with cover crops. But why do they not only use leguminous crops that can fix nitrogen? Why do they also use grain crops?
It's going to go back to the cost. A legume cover crop is going to be much more expensive than if you just plant a wheat or a rye species. And also finding a legume that's adapted to your environment can also be challenging as well. Although hairy vetch seems to do extremely well, Paul and I have come to realize that it's one of those that's difficult to kill. But really, at the end of the day, it goes back to the cost of the cover crop seed.
Okay, thank you. I have one more question.
Yes.
So in the cover crop study with the seeding rate,
Mm-hmm.
is 60 acre the recommendation, the national recommendation?
Yes, that's going to be closer to what NRCS is going to recommend. I believe it's, Paul, correct me, but it's probably going to be different depending on what region you're in. I believe here in this area, we're closer to 50 pounds per acre. Seeding rate is what NRCS is going to recommend.
Oh, then it's great that you found that even at 30, the results were very comparable.
Exactly. Yeah.
And I can interject there, Katie. For small grains, NRCS recommendation were as high as 80, 90 pounds in West Texas. And through research over the last several years, they reduced that 40 pounds for a drilled small grain rate in West Texas. So maybe we did something they listened to.
Yay. Thank you, Paul.
Hi, Dr. Lewis. I also have a question about the cover crop project. So why do you think there was a timing, that 2-week difference? Why do you think that timing made such a big difference in those cover crop yields?
So, of course, you know, with increased biomass production, that's going to be greater water use, but also what's happening during that time is you're going into reproductive growth stages. So that cover crop, whether or not it's wheat or rye, is also putting a seed head on, and that changes the carbon to nitrogen dynamics of the cover crop. And so you may be influencing or increasing your potential net immobilization that's occurring from a nitrogen standpoint. And so you may have greater nitrogen limitations where you have greater biomass produced, or at least potential for immobilization.
Gotcha. Thank you.
You're welcome.
Hey, Katie, this is Reagan. I'm going to be that guy that just makes a comment.
Okay.
Because that always has to happen in the seminar.
Yep.
Really enjoyed the overview of a bunch of your projects and some that you're involved with. But I just wanted to say I was surprised with your PCA analysis of the different, yield versus POC versus carbon, et cetera. I expected some collinearity between those when you were first going through, changes in soil carbon and whatnot relative to your cover crop practices. And then you got to these changes in the permanganate oxidizable carbon and the enzymes. I thought, well, I wonder how much those align with each other. Is it just a matter of having more biomass out there? Is it a matter of, if you influence this, is it just automatically influencing that? But looking at that PCA, they didn't really predict each other.
Not at all.
Which was really exciting and interesting and confusing at the same time.
Yeah.
Oh, wow, that's kind of cool. So anyway, any thoughts on that? I would love to hear. But that one kind of caught my interest.
Yeah. So really the only place that we see a strong relationship is going to be between, I don't want to be that person that goes back, but where we see that strong relationship between organic carbon and active carbon. And we're surprised that we don't see more of a relationship. So that was definitely a good catch on your part. It is somewhat shocking that we don't see a stronger relationship between those other parameters.
Yeah, no, very cool though. I was excited to see that type of analysis shown.
It'd be really interesting because Paul also collected samples, but from the bushland area, from the cropping systems that they have, long-term cropping systems they have up there, to see how soil texture influences what's going on. Because he may see very different results. He measured all the same parameters, but in a more clayey, heavier textured soil compared to where we were collecting samples.
Cool.
Yeah.
Thank you.
Thank you.
Any other questions?
Dr. Katie, I had a question on the cover crop project as well.
Mm-hmm.
So while looking at the soil health parameters, we saw that there is a high soil organic carbon and other soil health parameters. The enzyme activity was good in cover crop, no-till practice, but the yield was less compared to the conventional practice. So So what is the reason for that? Is that like the soil health is good, but the yield is not getting the benefit of the soil? Or
That is
what's
an
the
excellent question that I don't feel we necessarily have the answer to yet at this point in time. It's something that we're working on, and that's why I mentioned, you know, there are several different aspects. Is it a nitrogen or possibly phosphorus limitation in these systems due to immobilization? We have inherently low organic carbon levels in our soil. And so when we're adding, you know, 4,000 to 5,000 pounds of organic material to these systems, obviously it's going to be influencing the biological activity in the soil. And so I think that's why we're seeing those improvements. Why it's not being reflected in yield is a question that we are yet to find the answer for.
Yep. Got it. Thanks, Dr. Casey.
Thank you.
Well, you did an excellent job, Katie. Once again, I'd like to thank you. We had good discussion
Thank
on
you.
this.
Yeah, no, I'm impressed with all the questions. Thank you all.
Exactly.
Yeah.
And so I think we're done for the day, and we'll pick it up next week with another great speaker. See you
Awesome.
then.
Thank y'all.